So I'm going to tell you about protein nanopores, which are a really sort of intriguing entity to do basic research on, but which have also proved to be very useful in biotechnology. So let me just start by telling you what we're talking about. So I'm going to talk about protein nanopores, which are a few nanometers in diameter, and you'll see why that's important later. So the needle that you would have your COVID jab with will be about 500 microns in internal diameter, and the human hair about 80 microns. And then for the scientists that are listening, blood capillary about 5 microns, some filters, nuclear pore filters, 20 microns, pores in the nucleus of the cell down to about 30 nanometers, but we're talking about entities that are quite a lot smaller, and they will have about 1/50,000th of the diameter of a human hair. And as I'm going to tell you, they have interesting basic science and very useful applications in biotechnology. But I thought I'd begin about telling you how I got to Bayley, went away, and came back to Oxford again. So I was brought up in Prestatyn in North Wales, which used to be in Flintshire, but was mysteriously moved to Denbighshire while I was in the States. And this is my brother here, here's me. My brother chose to be a poet, which I think is probably maybe a little easier, I think, in retrospect than being a scientist. And Prestatyn was, my house was on the beach, and we had a place called the Royal Lido, and I remember the Beatles playing there in 1962, just after Love Me Do came out, which was their first record, didn't do very well. And anyway, they left, and subsequently I left and ended up in Oxford, and I haven't been back there for quite some time. So at Oxford, I came to Bayley to read chemistry, as Dermot told you, and spent a lot of time in the Dyson-Perrins lab. And I don't remember much from my Oxford interview. In fact, the only thing I remember was my question from Malcolm Green. And basically the question was, if there's a big pond with a pole in the middle, and I gave you a rope, how would you get the pole out? And I did manage to answer that question. I can't give the answer away because I believe it's still being used in Balliol interviews, but that's what got me into Balliol. And then, as Dermot said, I was very fortunate to have three terrific tutors, Malcolm himself and John Jones in organic chemistry, and David Turner in physical chemistry, the inventor of photoelectron spectroscopy. And when I made this slide, actually a couple of years ago, I couldn't find a photograph of David on the web, and I tried again yesterday. He's a really elusive character, but he was my third tutor. So after I finished my part 2 year with Jeremy Knowles, with the immortal words of Malcolm in my ears, "America isn't what it used to be," I set off to the States with my part 2 supervisor, and actually did my PhD at Harvard with Jeremy Knowles, and then moved on to do a postdoc at MIT with Gobin Khurana. And then I spent almost 30 years in the States. I was on the faculty at Columbia University, then the Worcester Foundation, Texas A&M University, and finally in 2003, when the CRL was put up, I came back to Oxford. All right, so how about a bit of science? So protein nanopores are found in membranes. And membranes are entities that divide cells into different compartments. So here's a nucleated cell here, and you can see there are mitochondria, the nucleus, endoplasmic reticulum, and so on. And all these different compartments are bounded by membranes. And these membranes contain proteins, and these proteins do a large number of different things. They can transmit signals across the cell membrane. They can transport things out of cells, waste products that cells don't want, or they can transport molecules that the cells need into cells, and have various other activities as well, lots of membrane-bound enzymes and so on. So the nanopores that I'm going to talk about are a special class of membrane proteins, and I'll show you a EM picture of a nanopore from Streptococcus here. This is actually one that is has quite a large diameter of about 30 nanometers. In this case, I'm going to talk about much smaller ones in a moment. And this is just sitting here in a lipid vesicle. And these nanopores, or pore-forming proteins, are actually used by us. We have a protein called perforin to kill off invading bacteria, or in this particular case, the bacterium uses these nanopores to attack our cells. And you'll see how the basic science of these proteins, but I won't stress that today, is very interesting. And what I will tell you about is their applications in biotechnology. So our favorite protein that you'll see several times in this talk is called alpha-hemolysin. In this case, it comes from Staphylococcus. It's a very simple protein. It's just 293 amino acids, and it's made as a water-soluble monomer, and it assembles into membranes to form a pore that has 7 subunits. So you can see the crystal structure here, and here's an AFM view of this mushroom-like assembly where you can actually see the 7 subunits about the central axis of the pore. So I think one thing that's very important to me is that our work really began as a basic science problem. So Oxford Nanopore and the ability to sequence DNA and so on really stemmed from our interest in the basic science of these pores. And the question that we asked originally was, how can a water-soluble monomeric protein assemble on a membrane to form a pore that goes through the lipid bilayer? And as you can see from this little sketch, we were able to work that out eventually. But the important point to me is that advances in basic science really very, very often come through discoveries in basic science. And second, that science is often highly multidisciplinary. So I'm going to tell you today about how ultra-rapid DNA sequencing was developed, partly in our lab and to a large extent at the company Oxford Nanopore, and also our recent work in using 3D printing to print synthetic tissues. And you'll see that these protein pores are involved in both of those activities. So I would stress that not only basic science is important, but interdisciplinary science is important. Now, I'm in a chemistry department, but to get all the things that I'm going to tell you about to work, we really had to use a bit of biochemistry as well, cell biology, some genetics, physics, a little bit of low-level maths, and some engineering to build some equipment. So we would not pretend to be the world's expert in any of these areas, but what we try and do is bring all these different areas together to solve specific problems in our lab. So to move on to what we actually do from the experimental side, there's one measurement that's very important, and this will apply to at least the first half of my talk. So we do a lot of so-called single-channel electrical recordings in lipid bilayers. So you can put a single copy of a nanopore, maybe the hemolysin pore, as shown here, into a membrane. And we do this in an apparatus that where we can form a little lipid bilayer across an aperture. And then we can apply a voltage, a potential, and a current will flow through a single copy of the pore. And that current is not carried by electrons as it would be in an electrical circuit, but it's carried by ions, say sodium and chloride. And we normally just call this an ionic current rather than an electrical current, although we do call this electrical recording. Now, when something happens inside that protein, and that's the whole basis of sensing with these pores or DNA sequencing using them, that current will change. And it's the modulation of that current that allows us to detect things that bind to the pore, that to detect chemical reactions going on in the pore, and to detect polymers such as DNA, RNA, polypeptides moving through the pore from one side of the membrane to another. So these currents are very small. Typically, we'll measure about currents of about 30 picoamps, but we can measure changes of those currents using very low noise amplifiers down to about 1%. That'll be important for sensing and sequencing, as I'll show you later. And this current is about 1/10,000,000th of the current in an old-fashioned light bulb. So here's our protein again, and I just want to stress that the dimensions of this protein are on the nanoscale, so that's why we call it a nanopore, even though I'm in angstroms here. But this barrel that goes through the lipid bilayer is about 2 nanometers across. And the important thing is that this is about the dimensions of a lot of molecules that we're interested in. So when they enter the pore, bind there, or undergo a chemical reaction, or pass through it, we can see that happening by looking at the electrical current. So the first technology that we really invented was called stochastic sensing. So basically, we have a single pore in the membrane. I'm just showing 2 states of the pore here. A current is passing through it, as I told you earlier. And if something binds to that protein, and that can be a metal ion, it can be an organic molecule, it could be a little piece of DNA, anything that will be in an aqueous environment, at least, you can see these binding events. So this might be the current going through the protein, then there'll be a binding event, come off, another binding event, another binding event. And this is a stochastic process, it's a random process, but the frequency at which these events occur tell us how much of this substance we have in solution. And they also, these events provide a signature. The amplitude of these events tells us something about, helps us to identify this molecule. And there are other signatures, noise during the binding, and so on. So based on that, we developed stochastic sensing. I'll just show you one example of that here. And it gives an example of how sophisticated the engineered pores that we use can be. So we took the hemolysin pore, mutated it so that it would bind a cyclodextrin molecule. Cyclodextrin molecule is rather like a donut. It's just 7 glucose units in a ring. And the inside of that molecule is quite hydrophobic. So it provides a sort of grommet within the pore when it's bound. It's shown on the top right here. And if you take ibuprofen, Advil, Motrin, they're all the same thing, the anti-inflammatory agent, and put it in the solution while a current is going through the pore, the ibuprofen will bind inside the donut and block the current going through the pore. So you'll see a series of binding events. And interestingly, the donut is chiral because it's made of glucose subunits. So for the chemists amongst you, these are diastereomeric non-covalent complexes. And in fact, you can distinguish the R and S forms, the mirror images of this drug molecule. It's actually the S form that's active. So if you look at this trace here, it's about 50 milliseconds across here. And the larger amplitude events are the S form binding, and the lower amplitude events are the R form. This is a racemic mixture of the two. If you bought ibuprofen and boots, that's what you would get. So this is just to show you that we can sense molecules in this way. And this is an exquisitely sensitive method for detecting changes in these molecules to the extent that we can detect isomers, as you can see here. So based on that, we thought that if people had used antibodies and enzymes, engineered forms, they'd been very useful in biotechnology and continue to be. Might it not be the case that protein pores could also have useful applications? And on that basis, in 2005, I founded Oxford Nanopore, and the company's been around for a while, but even as early as 2010, people were kind of after us. And I always like this quote: Bayley speaks in the laconic baritone of a late night radio DJ. If he has an entrepreneurial streak, it's not obvious. Well, if I actually had the talent, I wouldn't mind being a late night radio DJ, but we chose to go the way of Oxford Nanopore. As Dermot said, this company was successfully IPO'd 2 weeks ago, and it's valued at about 4 billion. And has over 700 employees. So we have the last laugh really on Kevin Davis. So another thing I'd like to point out is that at the time the company was founded, Spike Wilcox was working for IP Group, which was a company that helped us get Oxford Nanopore off the ground. And he was at Brasenose with Graham Richards, who you'll meet in a moment. He was both an undergraduate and did a PhD at Oxford. And Gordon Sanghera, who was appointed the CEO and still is the CEO of the company, was a postdoc with Alan Hill, who of course is famous for glucose sensing tests that are used today by diabetics. So I would also like to mention, as well as my tutors, Graham Richards, who's, as Dermot said, did lure me back to Oxford. And Graham is also an honorary fellow of the college. And he was, in fact, a research fellow at Bayley from 64 to 66. And when I came back to Oxford, he was chair of the Department of Chemistry. And he shortly afterwards became chairman of IP2 to IPO, which is the earlier name of IP Group. And he was really influential in getting me to form Oxford Nanopore, because he'd been through the formation of spin-outs companies himself, and really brought that culture to Oxford. Graham's written a number of books. His latest one is called A Scientific Life. It's quite a short read. And actually, I would recognize that. I would recommend that. I think in a very subtle way, he describes a lot of the good things and the less good things about Oxford, things that we'd like to preserve, things that we'd like to change. And I think you should pick that up at Blackwell's, have a read. So not long after we'd formed the company, and the company was actually formed for sensing of small molecules, the kind of example that I'd shown you earlier, but we wanted to look really at biomedical markers. We decided to change tactics and take up a much greater challenge of DNA sequencing. And this was a kind of Jules Verne type idea that had been around for a while, that you could use nanopores to sequence DNA, although no one had really got close to doing that. And there are really two ways that you might do it. One is that you might take your piece of DNA and chop off one base at a time and identify those bases just as we could identify ibuprofen. And the other possibility was to get a single strand of DNA to go through the pore and read off the 4 bases as they went past some sort of constriction in the pore. So you would just read them off at the time, C, A, T, G, and then C, and then G again. And that's really the method that has prevailed, and I'll show you that in a moment. Of course, it turns out to be much less easy to read off the bases, because the difference between this method and this method is this base that's sitting in our reading head here will have neighbors, and these neighbors influence the current, the changing current that's brought about by this base. So you have to use much more sophisticated methods to deconvolute the electrical signal. So I'm not going to go through the whole history of nanopore sequencing, but let me just tell you that there were a few major hurdles that had to be conquered. You had to get the DNA to thread into the pore, and if you've tried threading a needle, it's not always the easiest thing to do. You have to identify the bases, and as I told you, it's not quite as simple as just reading off 4 different current levels. Had to get the DNA to translocate through the pore. If you do get the DNA to thread and apply electrical potential, it tends to go through at about 1 to 10 microseconds per base, which is far too fast to read the details of the electrical current. And then, as you'll see later, just having one nanopore would take you a long time to sequence a large genome. And one amazing thing that Oxford Nanopore did was produce arrays of pores that could be used to sequence many molecules of DNA simultaneously. Okay, so once all that was done, so we're skipping, gosh, many years there, but from just the years at Oxford Nanopore, it took 7 years to actually develop the MinION DNA sequencer. And so this required so-called patient investment. You didn't want to have investors involved in the company that wanted to turn it over very quickly. But the MinION device, here you see an early one that's sort of stuck together with a piece of tape. But the MinION device is a portable sequencing device. It literally is this small, and it's cheap, and it's actually free. It's the reagents that are at some cost. It can do ultra long reads, single reads of over 4 megabases, which is just extraordinary and something we didn't really predict at the time. And as I'll show you in a moment, the device contains about 500 working pores. And again, as I'll show you in a moment, the accuracy of sequencing has been greatly improved recently. Initially, well, in 2012, it was probably around 85 or 90%, and not quite ready for prime time in some ways. But recently, the accuracy has been taken above 99%. So this is how the sequencing works in the MinION. To get the DNA to move through the pore, you can use an enzyme, and nowadays we're using a helicase. A helicase is an enzyme that takes a double-stranded DNA, which is much easier to handle than single-stranded DNA, and in an ATP, energy-dependent manner, splits it into 2 strands and can feed 1 strand through the pore. And as it goes through the pore, you see this modulation of the current that's carried through the pore by ions, because we apply a potential across the membrane, and you can decode this signal to get the DNA sequence. So let me just tell you very quickly some of the improvements that were made over the years. So even if we go back 3 years now, the accuracy of the so-called base calling, just putting a single, just in a single read, was as low as 92%. But this was quickly improved over 2 years to 98%. And now it's actually about 99%. And that was done in two ways. One was by literally testing thousands of different nanopores, different mutants of various pores to see which ones perform the best, and also by huge advances in methods for decoding these electrical signals, which are fairly primitive at the beginning, but which are really quite sophisticated methods for identifying the bases nowadays. I mentioned that you need a chip that will at least have several hundred pores, and you might say, well, why is that? Well, we would like to read genomes very quickly. The cost of genome sequencing has been reduced enormously to less than $1,000 for a human genome. And so it's really speed that we're interested in at the moment. Human genome is huge, 6 billion bases. And at 10 milliseconds a base, it would take about 700 days with a single nanopore, 2 years, to get a sequence of the human genome. If you had 10,000 pores working in parallel, and you'll see in a moment that we're getting pretty close to that, you could bring that down to less than a day. And of course, the long-term goal is to get hundreds of thousands or 1 million pores working in parallel. And of course, the bacterial genome is much smaller and might be sequenced in a matter of seconds if you can sequence from many pores in parallel. So I think that the Oxford Nanopore chip is an amazing technical feat. So within this MinION sequencer is a chip that contains a couple of thousand apertures. So when I showed you the bilayer recording earlier, we just have one aperture in our apparatus that we use in the lab. This chip literally has a couple of thousand of them that you can see here, just stained with fluorescent dyes. And each of these chips are, each of the apertures in the chip can produce data of similar quality to an amplifier that will cost us between £5,000 and £10,000. So this is a very high performance device. And with 500 of these out of the 2,000 pores working, you can get about a gigabase of data every hour. In fact, that's improved to now. So basically, I'm showing you this slide because it probably cost Oxford Nanopore a huge amount of money to make, so I had to show it. But it basically shows again the sequencing, the helicase splitting the strand into two, and one strand moving through the pore, and the electrical signal is modulated as the bases go through. And the difference here is that we have many hundreds of these nanopores working in parallel. And when you think about it, each of these wells is separately electrically addressable. So it doesn't matter that if you put a whole mixture of DNAs, just a whole sheared human genome through this, because you're getting, you're doing a single molecule experiment in each of these wells. So you don't have to use PCR to amplify a particular part of the genome and then sequence it. You can sequence all these fragments together in parallel. So at this point, Oxford Nanopore have this parallel sequencing device. It's really developed pretty much in the open nowadays. Sequencing data, software improvements, new sample prep protocols are shared on the web, and there are about 17,000 people who have been contributing to this kind of community on the web that tries to improve the technology in parallel with Oxford Nanopore making its own improvements. And the MinION is used in more than 100 different countries. And up till August, there are about 2,000 scientific publications that include nanopore-based sequencing. So from historical point of view, told you Nanopore had this working, well, they announced it in 2012, it's working in maybe late 2010, 2011. But the first public release of data was from Nick Lohman's group, who's been a pioneer in using nanopore sequencing at the University of Birmingham. And this was, at the time, a very long read, over 8,000 bases from a pathogenic bacterium, Pseudomonas. And here you can see the readout from the sequencer. And here's the sequence on the left-hand side. But I think what's interesting was that this wasn't published in Nature or Science. This first publicly available nanopore sequencing was actually published on Twitter. And that was quite a while ago. I think we're seeing a lot more of this kind of activity now with all the bioarchives and so on. After that, nanopore sequencing was used for small genomes, viruses, 2016, 2017. Ebola virus, of course, there's no such thing really as Ebola virus. There are thousands of different Ebola viruses, and you can make a family tree of Ebola viruses, and Nanopore was part of that. And some of these are far more pathogenic than others. And Zika virus in Brazil. And then a couple of years later, as the technology improved, human genome was sequenced using nanopore sequencing. Highly repetitive sequences that can't be tackled by other sequencing techniques could be sequenced by using nanopores. And importantly, RNA can be sequenced directly. You don't have to convert it by reverse transcriptase into DNA to sequence it. And there's obviously a lot of interest in sequencing RNA transcripts recently. So what is the future? Well, the latest device that's being brought up at the moment is called the PromethION. This actually contains 48 chips or flow cells. And instead of having 500 pores, each of these has 3,000 pores working simultaneously. So this has about 300 times the capacity of the handheld MinION, and it has the potential to yield 15 terabases of DNA in 48 hours. I think they've got to over 10 terabases at the moment. And this easily enables a sub-$1,000 nanopore-only human genome. And each of these flow cells can be operated simultaneously, and DNA from different viruses can be tagged in various ways. And even using 12 of the 48 flow cells, 2,000 COVID genomes can be sequenced in one day using 2 runs of this machine. So this machine has the capacity to do almost 10,000 coronavirus genomes in a day. And in fact, at this point, over 400,000 coronavirus genomes have been contributed to the database by nanopore sequencing. At the same time, you can, Oxford Nanopore are developing tiny devices too. And here's one that's not been released yet, but it might just be clipped onto your mobile phone to sequence DNA of who knows what, some film star on the next table in a cafe or whatever. So can do very high throughput sequencing, but also very quick sequencing of say, a plasmid or bacterium. And just to finish the nanopore part, I just show you some of the again, some new developments. One is that the pore that's being used now is not the hemolysin pore, it's the so-called CSGG pore. And this molecule, just to, again, illustrate why basic science is important, this molecule transports polypeptide chains to the surface of gram-negative bacteria, such as E. coli, and enables the formation of biofilms, these sticky, nasty films that bacteria protect themselves with. And so it was, again, a piece of basic science that was going on. Oxford Nanopore tested over 3,000 mutants of this pore to find one that would carry out the improved sequencing that you can see nowadays. And another interesting development not released yet is this, what I call this intelligent sequencing, where you have this very long piece of DNA, you can run it through the pore and sequence it, but then you can actually reverse the voltage, pull it back, and read it again. So you can read the same strand of DNA over and over and over again. And that, of course, greatly reduces the error rate in the sequencing. So what of the future? I think the future of Nanopore is actually to look at proteins. As you would know, the human genome, despite the 6 billion bases, only contains about 20,000 genes encoding proteins. A somewhat larger number of transcripts is made in most cells from these genes. But interestingly, many different so-called proteoforms, different forms of proteins, are made from these RNA transcripts, and they can be modified after translation. The transcripts can be spliced before they're translated and so on. And this in such a way you end up with this huge number of proteins in cells, and the nature of these proteins cannot be predicted from the genome. So one of our aims at the moment is to count the various protein species in cells, map these modifications, and remember, this is a single molecule technique. We would like to be able to do this at the single cell level. Well, next I'm going to tell you about how protein pores are involved in making synthetic tissues, but I just wanted to have a few slides on one of my favorite things, is using nanopores to look at covalent chemistry. So this is really just for the chemists that are out there, but it's you often get to talk about things that you're pretty interested in, but sometimes not about the things that are in the top of your mind at the moment. But as well as detecting small molecules and sequencing DNA, you can also look at bond making and bond breaking reactions inside nanopores. And I'll just show you one example here. So this is a complex reaction network. It doesn't really matter what it is. It involves substitution of an organoarsenic compound. But the important thing is that there are 38 possible reactions that can go on here. And because this is a single molecule experiment and each of these transitions is separated in the time domain, you can measure the rate constants for all of these reactions. And to cut a long story short, the green ones happen and the red ones don't. And that allows you to deduce something about the chemistry of substitution reactions at arsenic. And the kind of signal you get looks like a kind of new age music score. So these are the different current levels that you see, and you can see the interconversions of these different species in this scheme. And some of these happen and some of them don't. But actually, by looking at all the time intervals in this kind of trace, you can actually get out the rate constants as well as see what is allowed and see what isn't. And very quickly, another thing that we've been doing recently is making little tracks inside our protein pores, and we can actually watch molecules walk up and down one step at a time. And of course, we're interested in this because we can use this for chemically stepping DNA rather than using an enzyme to do that. And so we can get DNA to move down the pore when it gets to the bottom, change the voltage, flip it around, and get it to move up in the other way. And the chemistry for that is very simple. It's just the DNA is connected through a disulfide bond, and then a free sulfhydryl group comes along, forms a new disulfide bond, breaks this one, and then passes it on to the next one, as you'll see in this very quick movie here. Bond is broken, flips over, another bond is made, it's broken, flips over, and so on. And that's how you can make a very simple walking molecule compared to some of the things that are out there in the literature. Okay, so let me finish, probably take me another 10 minutes, by first of all showing you another piece of basic science, but this was the basic science that's led to what we're doing in the lab now, which is using 3D printer to build synthetic tissues and also print living cells. So we wanted to miniaturize those electrical recordings I showed you earlier. Normally, we'd have about 1 ml of solution in each chamber, but we found we could use small droplets, a couple of 100 nanoliters, in an oil. And if we had lipid molecules in the oil, a bilayer would form at that interface, a membrane would form that interface, and we could get a protein to go into that membrane, and then using these electrodes that you can see here, measure the current that flowed through the protein, so that the volume of these droplets are thousands of times smaller than the chambers we were using. So this allowed us to use very rare protein samples that we purified, or to use very expensive substrates. That was the whole idea. But then we had an even better idea that maybe we could take many of these droplets and have them interact with each other in 2D or 3D and form something that would resemble a tissue. What do I mean by a tissue? Well, if you look at a tissue in an organ in your body, at least one that doesn't have much extracellular matrix, you can see a whole bunch of cells that are connected to each other, and they interact with each other and the environment, obviously. And this kind of material has emergent properties. It's greater than the sum of the individual compartments. And what we've done is use 3D printing to make synthetic tissues, to print cells, and then also to make hybrids between the two. And to cut a long story a little short, we have our long-term goals are really to be able to use these materials, for example, to deliver therapeutics, and also maybe to use them to repair organs instead of doing transplants, for example. And one important thing, I think, when we've developed biotechnologies is to have them at very low cost. So, for example, human genome sequencing, when we started, Oxford Nanopore would cost about 20 or 30 million to get a human genome sequence, and it's down to a few $100 at the moment. And we'd like to be able to do the same with synthetic tissues and printed cells and so on. We actually got this idea from reading the Economist magazine. So I think Jeff Carr and the anonymous colleagues at the Economist had a couple of articles in 2011 saying that you could print a violin or print a chainmail glove. So we thought, why can't we print tissues? And basically, we've done that by printing aqueous droplets. They're about, in this case, about 50 picoliters or less in volume. And these droplets are separated by lipid bilayers, so they can communicate with each other with engineered protein pores, as you'll see in a moment. And we can add to them any functional components that we wish. So basically, I had a very smart graduate student from physics in the lab who built a 3D printer. You can't do this with a commercial 3D printer, to print these droplets, as you can see here. And you could end up then with various materials. And his one is printed in 20 layers and shows us an arborizing pathway through this little piece of printed material. And here's another example where something, if you look at this object in the X, Y, or Z direction, you can see a different pattern, and that gives you some idea of the fidelity of the materials that we're printing. If you think about it, these are not just packed spheres. These droplets will change shape once they pack, and in fact they're dodecahedra. And here's a rather nice photograph of a fragment of material that we printed and then polymerized and then taken this 3D image. So there are many things we'd like to do with these materials. I'm just going to show you one or two. One is that we're interested in signal processing by these tissues. So we'd like to be able to send a signal in, have it transmitted and processed in some way, and then have some sort of output. So for example, we might want to transmit electrical signals to living cells. We might want to have a material that can change shape, have a sort of muscle-like activity, or a material that can actually synthesize and release drug molecules. So just one of those examples is electrical signaling. So here we've printed in 3D this block of so-called synthetic tissue. In the droplets that contain the green dye, we put our alpha hemolysin pore, and that allows the interfaces between the green droplets to conduct ions. And so when we bring up these two electrodes, these sort of green spheres here, and apply a potential, a current will flow through this pathway, and it won't flow through any other part of this material. So this is a different mechanism to how a neuron works, but it really mimics how a neuron works. It's the fast directional transmission of an electrical signal. And we've been able to make that, a little more sophisticated by actually making the protein pores inside these droplets, and a little bit of molecular biology here. But bear with me. We made a gene for our hemolysin pore that has a light activatable promoter. So this actually contains chemically modified DNA bases that contain the so-called biotin molecule at one end. So it'll bind a big protein that will actually block transcription of this gene. And then when we shine light on it, there's a photocleavable group, and this big protein is released, and you can now transcribe the RNA, which is ultimately translated into protein inside of our synthetic tissues. And this just shows the kind of outcome here. Here's a piece of printed tissue. It contains a light activatable gene for a yellow fluorescent protein. And we've shone light on this, and this protein has just been made in selected compartments in this tissue. And then we can bring up electrodes just like we did before and transmit an electrical signal through this block of material. So those are synthetic tissues. Related to that is our ability to print living cells. And we've used really a very similar technology to put living cells into droplets and print them into various patterns. And this is a really, really exciting area at the moment, given all the things that are happening with stem cells and genome edited cells, and using patients' own cells in diagnosis and in medical treatments. So again, rather quickly, living cells can be patterned by a similar droplet printing technology. So we print these originally in oil, and then we transfer these printed entities into growth medium, and the lipids that surround these droplets originally then get eaten by the cells, and we end up with a patterned piece of tissue. And just to show you an example here we've printed neural stem cells, and after 8 days in culture, you can see they're putting out axons, processes, and 14 days of culture. If you look at the high magnification at the bottom, these are really beginning to look like neurons. So these printed cells have very high viability. They're all alive. They can be pre-patterned in various ways. They're functional. We can show electrical signaling in these neurons, and they can differentiate and migrate in sort of biologically appropriate ways in these printed materials. And one project that we've started on recently is sponsored by the Oxford Martin School and involves our lab. Lena's a postdoc in the lab, and Francis Zeller and Zoltan Molnar in DPAG, Physiology and Anatomy and Genetics Department. And we're trying to print layers of the cerebral cortex, and then these can be used to repair a damaged brain initially in mice. And again, finally, we can make hybrid tissues. So we would like to be able to link our synthetic tissues to printed cells, to living organs. And one way that we're doing that is to use our favorite protein, the alpha hemolysin. And we've been able to take that protein. Remember, it's a heptamer, 7 subunits. So we've been able to make a dimer of heptamers. So this is the mushroom cap on the mushroom cap. So it's got 2 membrane spanning domains, and we can take this engineered protein and allow it to span the membrane of our synthetic tissue and span the membrane of living cells, and thereby connect them electrically. And here you can see this experiment here between 2 lipid vesicles, these dimerized proteins spanning the lipid bilayers in both of these liposomes. So in the new work in our lab, we can make both synthetic tissues and cellular tissues by 3D printing at high resolution. We can get them to communicate with each other, and we're trying to explore their fundamental biology and apply them in medicine and surgery. And I'll just end up by sort of recapping the kind of history. Really, it just started off with an interest in membrane proteins. It was a basic science interest. One of the problems was, how do pores assemble from a water-soluble form into a membrane? That led us on to doing single molecule sensing, which led to the formation of Oxford Nanopore. And Oxford Nanopore, building on the academic science, brought single molecule DNA and RNA sequencing into being. At the same time we were doing this, we wanted to miniaturize these electrical recordings. So we used these very small droplets, nanoliter droplets, and that led us to printed droplet networks in which the droplets can communicate with each other and at the outside world. And that in turn led to synthetic tissues and printed cells, which has led to a new company called Oxybio. So if there's any overall message, it's keep doing basic science, because it will lead to all sorts of interesting applications. And I'll just end by thanking our sponsors and people in my group, some of them very young, and thank Bayley. And thank you very much for listening.